It's 5 p.m. So hello everybody, and welcome back to a new session of the EDIT Sickle Cell Disease webinar program. Let me introduce myself first. I am Alba Albert, Scientific Manager of ERN-EuroBloodNet, the European Reference Network for Rare Hematological Diseases. And I am very happy to welcome you to the third webinar of this series of 7 webinars that are designed for people living with sickle cell disease to provide an overview of gene therapy and genome editing approaches for the new therapies for this disease. And session 1 was an introduction, and the session 2 was centered into the basics of gene therapy with the lentiviral vectors. And today in session 3, we will revise genome editing, CRISPR Cas9 technique, and how it works and its uses. So before starting, I will just point out some of the housekeeping notes that are just for you to know. Your microphone it's muted and will remain muted during the presentation. Note that this video is recording, and all the videos will be implemented in the e-learning environment of the ERN-EuroBloodNet and the EDIT Sickle Cell webpage. And please submit your questions via the Q&A chat box at any time, and we will answer them in the second part of the webinar. Finally, just a reminder on how to turn on the Zoom AE translation captures. You have to click on the CC enclosed in a square in the bottom, in the Zoom toolbar at the bottom right hand side, and then select your speaking language as well as your caption language of interest. Having said this, I am very happy to introduce the speakers for today's session, that are Annarita Miccio from the Imagine Institute in Paris and director of the EDIT Sickle Cell Disease program, Claudio Mussolino from Universitaetsklinikum Freiburg, and Miriam Amendola from the INSERM, also based in France. So thank you very much, and I hand over to Claudio that will start with the first part of the webinar. Thank you.
Thank you very much, Alba. Very much pleasure for me to join this webinar. I see now the slides. Okay, so then, so first of all, we have nothing to disclose, and no specific things to disclose here. Okay. So as a first, I would like to give you a little bit of a general overview of what we will be discussing today. We will, of course, I will give you a very brief recap of what you have heard in the previous seminar. That will be very, very brief to don't bother you with the things that you have already heard of. And then we will dig a little bit deeper into what are the options, or what does gene editing mean for, let's say, for the sake of gene therapy, of correcting specific mutations. And then I will give you a little bit of a feeling of how this gene editing works to correct mutations that affect our body. And we will then speak a little bit about the different tools, and then we will see how this is particularly applied to the field of sickle cell disease, how we try to do that to develop a therapy for this devastating disorder. And then we will go through more the clinical aspect of what has been approved, and what you are, I'm sure, aware of, the Casgevy product, and what are the challenges of gene editing. We will discuss, we will cover all these different topics. There is some pop-up window, sorry. Okay. We will go through this all these different topics, and so we'll start now directly with giving you a brief recap of the last webinar. So you have heard of ways how to potentially treat specific genetic disorders. And you know that you might have heard that the way gene editing works is trying to directly correct mutations within the genome of a cell. So now we can do this in different ways. We can think of having a gene which does not work in our cells, so it does not do the job that it should do, a mutated gene, as you see here. And one easy concept to develop a therapy in this case is to just provide an additional gene which has the very same function, which is, let's say, the correct function. And so we just could transfer now this gene within our cells. So our cells will still contain the mutated gene, but on top of them, we'll also have a new copy of a corrected copy of the normal gene that would make its function. So this is a way which is typically goes under the name of gene addition strategy, in which we typically use different technologies to just provide the cells with the gene function which is missing because of a mutation. However, as you see there on the picture, where the mutated gene is still present, we just add the normal copy of the gene on top of that. And with the gene editing technologies, we try to be a little bit more precise. We try to directly correct the specific mutation which is in the cells to just revert it to again normal sequence. And so we go rather on the very root of the disease, and we try to correct a specific genetic defect in order to restore again the normal function of the gene. If you go to the next slide, so we can then talk a little bit of how this gene editing procedure works. Basically, if you go to the next slide, the concept of how, so where the gene editing moves from is the ability of targeting very specific sequences in the genome, where the mutation that we want to correct is located. And to do that, we need something that allows us to detect the sequence where the mutation is, and then to like molecular scissors, try to cut the DNA molecule in that specific area in order to replace the wrong sequence with the correct sequence. And so, in a way, correct precisely the mutation which is underlying that specific defect. So in this case, or what you see here in this picture, if you imagine that you have a very specific mutation in your genome, so we need something that allows us to detect that specific sequence, and we need another component that allows us to cut specifically that DNA in order to replace this wrong sequence. If you go to the next slide. We can try to understand a little bit now how this technology works. In general, these two functions, the detection and the cutting, are performed by two different technologies. On one side, we have a DNA targeting domain. How is it called? So something that allows us to specifically reach sequences on the genome. Our genome is very long inside the cells, it's packed in the nucleus of each cell, and it's about 3 billion of nucleotides. And so among these 3 billion of nucleotides, we have to be sure and precise in detecting only, or in identifying only those which are mutated and that we want to change. So we need a technology that gives us the power to search this very long sequence of nucleotides and find those that we want to target. And then we need an additional technology that you see here indicated as a nuclease that allows us to now perform a cut at that specific region, because after that we perform a cut, we can use basically use the normal mechanism that the cells would use to repair the DNA, but use it in order to correct the specific mutation, and we will see in a minute how. These two components, the DNA targeting component and the nuclease component, are linked together in a single molecule. So now this is how the old molecular scissors work. And I'm sure you have heard a lot in the last maybe 6-7 years about the CRISPR system, why this has been so much a game changer in the field of gene editing. So if we go to the next slide, this is basically indicated here. So the power of this novel technology, the so-called CRISPR-Cas9, stands in the fact that this system is extremely easy to handle. While with the old technologies, every time that we had to change the location where we want to correct a mutation, we had to produce a new protein, a new detecting technology that could be targeted to the new sequence that we want to target. In this case, with the CRISPR-Cas9, the targeting to different location is much easier because this occurs through this component that you see here, which is called guide RNA. And basically, the guide RNA can be changed very easily by, let's say, just buying, let's put it that way to make it very simple, just buying a fragment of RNA which contains the sequence that we want to target. And so this makes the ability to target different sequences in these 3 billion of base pair of our genome very easy. So with this technology in hands now, we can very easily target different regions in our genome, and this allows us to be very flexible in the way how we can target a specific location, as for example in the case of the sickle cell disease, and to correct the mutation of causing sickle cell disease. So if we go to the next slide, in the case again of the sickle cell disease, what we can think of to correct this specific disease. So now we start with the cells that have a mutation in a gene, which is the beta globin gene, and this gene now is producing an information which is not correct. And instead of producing the healthy hemoglobin, which is that molecule that is supposed to bring the oxygen to all our tissues and all our cells, this hemoglobin, which is called the sickle hemoglobin now, is not able to do so. And since this hemoglobin is loaded in some specific cells in our blood, which are the erythrocytes, this assume this sickle shape because of this mutated hemoglobin. And this is, as I said, these cells are no more able to do their normal function. So now there might be several ways to develop a therapy for this type of disease. On one side, so based on what I have told so far, we have in our hands a platform that allows us to detect the sequence which is wrong in the beta globin gene and revert it to normal. So we can correct the mutation directly in the site of mutation in the beta globin gene. On the other side, what we can do, we can try to reactivate another gene, which is the hemoglobin gamma gene. And this is basically an alternative to the beta globin, but it's just more active during the phases of our development, while let's say the fetus is still inside the body of the mother. And in that phase, let's say the hemoglobin that is produced by this gene is still functional in normal situation. But when we born and then we become adult, this gene is silenced, so it's inactivated because the function that it does, it's replaced by the function of the beta globin gene. And so, of course, now if we have a mutation in the beta globin gene, but we do not have a mutation in the gamma globin gene, in principle, we can revert now, and we could reactivate the gamma globin gene so that also in the adult phase, this gene could, in principle, produce now the fetal hemoglobin, which could be still as active as the normal hemoglobin and then restore the function. So now there are these two ways of developing a therapy for sickle cell disease. And if we go to the next slide, we can a little bit try to understand how we can develop the tools for correcting now this type of, or to apply this type of therapies. As you see here, so this is the situation now that we would find in the DNA of cells which harbor the mutation leading to a sickle cell disease. So in the coding region, in the region of this gene, of the beta globin gene that produces the beta globin, there is a change in a single nucleotide. As you see here, this G, A, and G, these are nucleotides that compose our DNA. And in one of these mutations, this A here is changed to a T, and this produces a change in the protein, which eventually leads to a protein which is not functional. And let's say what we can imagine now to make this the things a little bit less complicated is that we have the information in the gene that could produce hemoglobin. However, this information now is wrong. It produces something different because it has this little change in its sequence. And you see, like here, this is exemplified, for example, by this sentence: "The fat cat ate the rat." So this is the original meaning of the sentence, in which there is a fat big cat who is able to eat a rat. But if we now generate a mutation and we change this sentence to "the cat ate the rat," this doesn't mean anymore anything. And so this is the idea how now a single change, a single nucleotide change in the genome can change so much the function of a protein, which then leads to a protein which is non-functional anymore. And so what is our goal now with the technologies that I discussed so far is to try to revert this type of mutation and trying to correct it, to establish a novel class of therapies that is based on directly editing or changing the genome which is inside the cells. So if we go to the next slide, we'll look a little bit more on how this genetic surgery works, basically. And you can see here exemplified in this slide. So if we consider now we have a gene in our body that has a certain information, and maybe the information that you see here is what is written down here: "Mary walks alone in the dark." So this is basically something that is can create, concern. So a little girl walking alone in the dark. So it's something which might be not an information that is a correct meaning. So what we could do here, for example, is to target this information and just destroy it. So just introduce some changes that would just change the meaning, in order to avoid that this information is delivered to the cell. So we don't want that Mary walks alone in the dark. And so what we can do, we can use CRISPR-Cas to target this sequence and generate some random change. You can see here, for example, we can generate some insertions. So what the CRISPR-Cas can lead to the introduction of additional letters in that sentence, so that the meaning is no more there. The meaning is disrupted. And so Mary will not walk anymore in the dark, because the meaning is wrong. As you see here, Mary P walks alone, P in the dark. So there is no meaning. An alternative way that can also happen is that as a consequence of the targeting of this sequence through the CRISPR-Cas9, we can have, instead of insertion of letters, we can have deletions of letters, but the result is the same. So the meaning of the sentence is disrupted. And so now if we translate now this concept a little bit to the context of sickle cell disease, and this will be discussed a little bit later, we can use CRISPR-Cas to inactivate informations that lead to the inactivation of gamma globin, which, as I said you before, is a gene that we have still in the body, but it's switched off, it's not active. So because there are mechanisms that keep this gene switched off. And so we can tell through the CRISPR-Cas system, we can tell to our cells to block the information that keeps beta globin inactive. And so then this would lead to the reactivation of the gamma globin gene, and so to the production of the fetal hemoglobin. So this could be one way to use the CRISPR-Cas to inactivate some information that we don't want, basically. If we go to the next slide, we see rather how we can use the CRISPR-Cas to correct a specific information. For example, again, we have this gene that produces that has a certain information. Here you see Mary walks alone in the park. So now this is the correct information and not Mary walks alone in the dark. And so we can consider that now this single letter change, dark and park, let's say this is the mutation that we have. And we can use CRISPR-Cas to now change again back that D of the dark back to a P of park. And what we have to do in this case, we have to get into the cells with our genome editing technologies, also with the CRISPR-Cas. The CRISPR-Cas will recognize the area where this information, so this wrong information is stored. And if we now provide also a piece of DNA which contains the correct information, then we can use this, or the cells use this additional piece of information that we are giving from the outside, so a piece containing the letter park, and the cells will use this to repair the broken information that we have targeted with the CRISPR-Cas, and then is able to convert dark into park. And so now we again can get from an information which is not safe, let's say Mary works alone in the dark, towards a correct information which is Mary works alone in the park, which is something that we would like that happens. Okay, so we have seen now two different ways of using the same CRISPR-Cas. On one side, we prefer to just inactivate the gene because this drives a wrong information, it drives a dangerous information. So we prefer to inactivate it. And so this is one way of using CRISPR-Cas. So just create changes which inactivate the basic information which is there, because we believe that inactivating sometimes it's less dangerous than keeping that information. So we believe that better that Mary doesn't work alone at all, instead that she works alone in the dark. In another type of therapy, we can think of precisely correct the wrong information. So we don't want that Mary works alone in the dark, but we would like that she works alone, okay, but in a park, so in a situation which is less dangerous. And so we can precisely correct this information in the genome with the very same tool that we have seen before. And if we go to the next slide now, I will now hand over to Annarita, that will tell you a little bit more of how these technologies are then used into the context of sickle cell disease. Thank you very much for listening.
Hello, can you hear me now?
Yes, Annarita.
Okay, sorry. So thank you very much, Claudio, for this nice introduction of the CRISPR-Cas9 system. So now I will try to give you an idea of how we can use this CRISPR-Cas9, so this genome editing system, to develop therapeutic strategies for a patient affected by sickle cell disease. So in particular, we will discuss about the Casgevy gene therapy, and I will just briefly remind you what it has been discussed so far, but also in the previous webinar. So as you know, in the adult life, so in the adult red blood cells, we have mainly the expression of the adult hemoglobin, or HbA, that is composed of 2 beta and 2 alpha chain, while in the fetal life, we have the expression of the fetal hemoglobin, or HbF, which is on the contrary composed of always 2 alpha, but also 2 gamma globin chain. And so the idea of the therapy based on Casgevy drug product is to basically reactivate the expression of the fetal hemoglobin that indeed in this patient can compensate for the defect that are affecting the adult hemoglobin, such as, for example, in sickle cell disease. So basically the goal of this therapy is to reactivate the expression of the fetal hemoglobin, but how to do it? So the idea would be to basically inactivate through the mechanism that Claudio just explained a gene that is called BCL11A. This is a factor that basically turns off the expression of the fetal hemoglobin. So it is a factor that inhibits its expression. So the idea is that if we remove this factor, then fetal hemoglobin can be produced again in the adult cells. And so if we go a little bit more in detail, so this is what happened in the adult cells in which BCL11A is this factor that basically binds to the gene that produces the fetal hemoglobin and inhibit the production of the fetal hemoglobin. So this is the situation that we normally have in our adult cells, but now with the CRISPR-Cas9 system and the Casgevy therapy, basically we inactivate this gene that encode for this inhibitor by basically cleaving into this gene and generating deletion and insertion. So if we inactivate the BCL11 gene, there is not anymore the BCL11 inhibitor factor, so BCL11 can no longer inhibit the fetal gamma globin expression, and so we have the reactivation and the production of the fetal hemoglobin. And so at the end, the final goal is to genetically modify the hematopoietic stem cells that finally will be able to produce red blood cells that carry the fetal hemoglobin. And so basically this is quite a smart therapy because actually Casgevy is able to inactivate BCL11 only in red blood cells. So again, it inactivates the factor, and so we have the expression of the fetal hemoglobin. This is very important because BCL11 is a factor that is present in many different cell types. For example, in the immune cells, that are other cells that are produced by the hematopoietic stem cells. So these are the white blood cells that are necessary to fight, for example, infection. So in these cells, BCL11 is very important. So we need to maintain the production of BCL11 in immune cells. And indeed, how do we do it? Basically, because when we use the CRISPR-Cas9 system, basically we cleave and inactivate only the genomic region that is important for the production of BCL11 in erythroid cells, while we do not touch the region that is important for the production of BCL11A in non-erythroid cells, so in immune cells. So for this reason, as we inactivate BCL11 only in red blood cells and not in all the other cell type, we can say that this strategy is quite safe. So basically, now I will briefly discuss about the journey that a patient need to undergo when they receive the Casgevy treatment, and also the limitation of this treatment and the comparison with other therapies. For example, the comparison with the gene addition therapy that were mentioned at the beginning by Claudio, and in the second webinar, the therapy that aim at just adding a new gene to the cells using a lentiviral vector. So this is the typical journey of the patient that receive the Casgevy therapy. So first of all, we have the collection of the hematopoietic stem cells from the patient. Each time it takes a few days, but it's often we need to perform a multiple collection of the hematopoietic stem cells to obtain a sufficient number, and therefore the patient can come to the hospital in several times, not only one time, up to around 5 times. And so this is why this process can last a few days, but mostly weeks, because we have to do multiple collection. Then the cells are genetically modified in vitro in the lab by administering the Casgevy treatment, so the CRISPR-Cas9, and this can take some days to months because we need to genetically modify the cells, but also we need to do very stringent quality controls to be sure that the therapy has been in these cells effective and safe. And then once the drug product, so the cells, genetically modified cells, are ready, the patient come back to the hospital and he receive a conditioning regimen. So he receive high doses of chemotherapy, as we discussed in the previous webinar, because we need to get rid of the mutant hematopoietic stem cells and to make space in the bone marrow for the newly genetically modified hematopoietic stem cells. And then the cells are the genetically corrected cells and then infused into the patient intravenously because this is the way in which they are injected. And then the patient remain for some weeks at the hospital until the immune system recovers. So this is quite a relatively long process. And for now, this is a treatment that can be proposed to patients usually 12 years old or older with a sickle cell disease, or with beta thalassemia, which is another hemoglobinopathies that affect the production of the adult hemoglobin, that can also be treated by reactivating the expression of the fetal hemoglobin. I just want to point out that, again, the patient receive a conditioning regimen, so a chemotherapy, but while in the bone marrow transplantation, the patient, as we see last time, they also need to receive immunosuppression, which means that they need to receive drugs that reduce the activity of the immune cells of the patient, because in the case of the bone marrow transplantation with a donor, an external donor, we need to reduce the activity of these immune cells that otherwise can attack the donor. However, the beauty of gene therapy, of this approach, such as the but also the gene addition approach using lentiviral vector, is that we do not need to give immunosuppressant because we are using the very same cells from the patient, and therefore this is much safer. But of course, there are risks that are associated with the Cas9 treatment, but the risks are quite well known and therefore can be mitigated. And probably most of the risks, they arise from the hematopoietic stem cell transplantation, not really from the gene editing procedure. So we have already discussed this risk for the allogeneic bone marrow transplantation and also for the therapy based on the lentiviral vector, so the gene addition strategy. So the use of a conditioning regimen, so a chemotherapy, can indeed be associated to risks such as infection or sterility. So infection, because it takes some time for the hematopoietic stem cells to then start producing again all the cells of the immune system that fight the infection, and sterility also because of the toxicity of the drugs that are used in the conditioning regimen. However, as I said, there are mitigation strategies. For example, in the case of infection, we can give a prophylactic antibiotic treatment, or in the case of sterility, there is a sperm and oocyte cryopreservation that is proposed. And finally, a last limitation could be the poor graft function. So the cells that we give might not be functional and completely corrected. And as mitigation strategy in this case, we try to give the cells a graft, so cells that are edited with very high efficiency, so a graft in which the majority of the cells are genetically modified and genetically corrected. And of course, also another mitigation strategy is the choice of the appropriate conditioning regimen that is able to get rid of all the mutant hematopoietic stem cells of the patient so that they cannot outcompete the genetically modified cells. And in general, we have to say that as for the other therapies, a positive management and preparation of the patient increase the chance of the success of this type of therapy. And so, of course, such as for each therapeutic approach, novel therapeutic approach, there are inclusion criteria and exclusion criteria. So the patient need to fulfill all these inclusion criteria. So in most of the cases, they have to be 12 years old or older than that. They have to have a diagnosis, of course, of sickle cell disease with the recurrent vaso-occlusive crisis. But this strategy is also proposed for patients with beta thalassemia, again, another hemoglobinopathies that can be treated by increasing the level of fetal hemoglobin. The patient needs also to be able to undergo hematopoietic stem cell collection and the conditioning with the chemotherapy, in which we typically use the busulfan, and they do not have to be pregnant. On the contrary, exclusion criteria are active infection or medical condition that make the procedure unsafe. So the contraindication to busulfan to the required procedure, of course, the pregnancies, but also concomitant disease other than sickle cell disease or beta thalassemia. And of course, another last point is that one exclusion criteria is also the inability to collect enough stem cells, which unfortunately could be the case in particular for a patient with sickle cell disease. And finally, my last slide, I would like to briefly try to compare the two strategies that we have discussed in webinar 2 and in this webinar. So in the webinar 2, we have discussed a gene addition strategy, and so the drug product is known as Lyfgenia, and this is a therapy that has been approved by the FDA in US, which means that this has been approved by the American regulatory agency. This is not approved in Europe. And so, and this is using a lentiviral vector to add a variant hemoglobin. Well, today we have discussed the Cas9 treatment that has been approved by both US and European regulatory agencies, so by both FDA and EMA, and is based on the CRISPR-Cas9. So both of them have advantages and limitations. Lyfgenia, as we've seen last time, it can induce insertional mutagenesis because of the integration of the lentiviral vector, and so potentially malignancies that have been observed in some patients, but I have to say that these were very few patients, and there was not the relationship between these tumors and the integration of a lentiviral vector was not demonstrated. Well, in the case of the Cas9, a potential limitation is the toxicity that can be caused by the cleavage of the DNA. Still, we are cleaving the DNA, and this can be toxic to the cells. So these are some limitations specific of each therapy, but there are also common limitations. So the toxicity due to the chemotherapy, we just discussed all the risks that are mostly linked to the condition and the chemotherapy, which is similar between the two products, and mostly also the high cost, 2 million for Cas9, more than 3 million for Lyfgenia, and the complex logistic. Indeed, by reducing the cost or simplifying the logistic would really be beneficial to achieve a larger clinical success. I have to say that to tackle all these challenges, there are novel approaches that have been developed that will be discussed later in this presentation and in the next webinar. So we'll give the floor to Mario Amendola that will discuss then the challenges of gene editing. Mario, are you there?
Yes, I was waiting for being unmuted. So hello, everyone. I'm happy to be here to present in this webinar about sickle cell disease. So my role here is to explain you the fact that this new strategy that we have discussed so far do not come for free, in the sense that as any other drug which has been developed so far and we use every day, there are some side effects. So these side effects are potential side effects. I will discuss them. So the idea here is to I mean, we have seen how powerful this tool is. The idea now is also to make you understand which are the reasons why it's difficult to develop this kind of strategy quickly and for any kind of disease, because we have a lot of safety study that needs to be performed in order to before this kind of treatment can be released to a patient. And these challenges that I will show you, they don't mean that the technology is not safe. It just means that before any treatment is approved, we need to undergo strict regulatory evaluation. So in these new slides, as I said, there are many different potential side effects that the use of CRISPR-Cas9 can have in the treated cells. Many of these potential side effects are very rare. Many of these potential side effects, as I told you, are potential, which means they could happen, but most of the time they do not happen. But I will just indicate which could potentially happen. And that's what's the role of the researcher, is actually first to identify which potential side effect could occur, and then try to actually measure them to see if they really occur or not. So the side effect could be there, but does not mean they are there. So there are many of different kinds. I will show you the main potential side effect that can happen when you try to play with the DNA modification. And there are two main kinds. So one, it's called undesired DNA edits. So you see that there is a target, and in the target by you, when you try to, if you play with the target, you can always go and target the on target, so the site that you want, which is in green, but sometimes you can also make a small mistake, and instead of aiming for the green target, you can aim for other target, which we call the off target, which are not the one that we want. And here are represented at the red cross. So which are these red cross? We try to modify sequence in the DNA, and these sequence are very specific. But sometimes, since the DNA is really big, you can find some sequence which are very similar to the one that you want to target, but they are not identical. So here you can see many different words, English words. In the green one, you can see to, and you can see, for example, other words which are very similar but not identical. So, for example, if you add an R, you have a word which is very similar, but it's totally different meaning. Or if you remove one letter, for example, if you remove one H, you have a word which is very similar, but with a different meaning. So sometimes if you try to go for the right sequence, but you can make a very tiny mistake, and instead of going for the right target, you can go for wrong target. And so you modify the sequence of the DNA that you don't want to modify. So this is one potential side effect. And another side effect I will discuss you afterwards. But just to be a bit more detailed, next slides, to be slightly more detailed about the off-target, I will take the same example that it was shown you before by Claudio using a sentence. So your on target is this precise sequence. So Mary walked alone in the dark. So this is the sequence that you want to modify, and you can modify, the aim is to modify a specific words in this sequence. But sometimes, as I said, the CRISPR-Cas9 can make a small mistake, much more rarely, but can still do them. And so you can find a sequence, so a sentence, which is relatively similar to the original one. So for example, it's Mary talks about her dress. So it's a sentence which is relatively similar to the one that we aim. And we can also modify a word which is similar but different from the previous one, maybe in a different location. And this is what is an off-target. So it's a modification of the DNA which you do not expect in a part of the DNA which is similar to the one of interest, but at the same time is different. So when you do this kind of modification, the risk is that you modify a different sequence in a different way. And so the outcome, so the final results that you obtain is totally different from the therapeutic one. So the risk, again, is that you have a totally different effect, but the luck is that this happens, if this happens much more rarely than the real modification. So what's the effect of this off-target modification? It can be very different. So sometimes this off-target modification is there, but it's not really important. So even if you modify this sequence, you don't really have an effect for the patient. Other time, this modification can have an effect on the patient. So and which is the effect will depend every time on the specific modification that you can have. So this is a very difficult side effect to assess, because first it's very difficult to find, it's very difficult to quantify, and it's very difficult to understand which will be the function. And so this is one first set of potential side effect that you can have when you try to modify the DNA sequence that you want. And then there is next slides. There is another side effect which is much more general. So when we use the CRISPR-Cas9, or at least the CRISPR-Cas9 that has been used in the Cas9 product, which is the original, let's say the original Cas9, what you do, you see that there are two scissors. So which means, and this scissor cuts each of the two strands of the DNA. So if you remember, the DNA is a double-stranded sequence. So when you use this CRISPR-Cas9, you cut both of them. When you do this kind of modification, this kind of alteration in the cells, this is very, very severe. So it's not a trivial damage that you induce in the cells. So this can be very important. And so for some cells like hematopoietic stem cells, which are the target cells in Casgevy, these cells are very sensitive. And so when you do this kind of DNA damage, you can sometimes induce some effect in the cells. So the cells can, sometimes some of the cells can die because of this DNA damage. Some of these cells can stop proliferating. And sometimes when you do this DNA damage, the cells try to survive, so they try to repair the damage, but sometimes instead of repairing the damage in the way that you would like, they can repair this DNA damage in different way, which can be sometimes toxic. And so that's why the researchers have developed new tools which can be safer than the original one. And so why they are safer? So there are different tools. I will not go in detail because there will be another webinar dedicated to this new strategy. But what they have in common, the base editing and the prime editing, is that you see only one scissor instead of two. So in order to introduce DNA modification, they do not need to generate a double-strand break, so they don't need to damage the whole DNA, but they do a damage in DNA, which is much more soft in the cells. So this is much easier for the cells to repair, and the cells suffer much less from this kind of DNA damage. And so that's why this new tool are now developed, because they are much more severe on the cells, much less severe of the cells. And so the cells will behave better after DNA editing. So if you go to the next slides, if you remember, when Claudio presented the possible way in order to treat the sickle cell disease, he showed you two main strategies. The strategy that we discussed the most and the strategy which is used in Casgevy is the reactivation of fetal hemoglobin. This strategy is based on the Cas9 system that we discussed. So you generate a double-strand break, and then you can reactivate the expression of HbF. And this is an indirect strategy. So you need to reactivate another gene in order to solve the issue. Much more straightforward would be the reparation of the nucleotide which is mutated in the disease. So you have a mutation that caused the disease, you simply correct this mutation. So even though this strategy seems much more straightforward, this strategy, in order to correct the mutation, it requires the delivery in the cells of a correct DNA sequence, which is used by the cells to correct the mutation. And this simple step, so the addition of correct DNA, is technically very difficult to achieve. So it's very difficult to deliver this DNA to the cells, and it's very toxic for the cells to accept this DNA. And so the problem is that you will cure the disease, but many of the cells will die. So it's very difficult to do this strategy. And that's why, so this is one of the other side effect, genotoxic effect, that we can have while we try to do this gene correction. And this is why today the most advanced strategy are actually based on the other technology, because it's much easier technologically to do. So in the future, potentially we will have both options, but nowadays the correction of the mutation remains still much more challenging. And so that's to explain you why sometimes we prefer one strategy rather than the other, because it's related not to the efficacy of the treatment, but to the toxicity, potential toxicity associated with one strategy rather than the other. So next slide. So for today, we will, the part of genotoxicity is concluded. We will discuss more about the safety and so the toxicity in the session 5 that will follow. We will also discuss about new tools that can be used to solve some of these issues. So ameliorate the safety, but also the efficacy, and this will be discussed next time in the session 4. And then we will see how this can be applied for the future treatment of sickle cell disease, which will be the next approach for this disease in the session 6. And then finally, in section 7, we will see how we can bring this new strategy to the clinic. So which is the regulatory path to go from the bench to the bedside of the patient. And next slide. And so this is just the acknowledgement. So Annarita Miccio, which presented today together with Claudio and other people that already presented in the previous webinar and will be participating in the next one. And then the people that helped us in preparing the slides and the graphic to show you the genome editing strategy that is available for sickle cell disease. So happy to answer to your question if you have any.
Thank you very much, Mario, and all the speakers for your clear presentation of today.
Thank you very much. I would just like to also acknowledge that the presence of Laure Joseph. She's a medical doctor at the Necker campus, that she's really the one who's treating patients. So she's also here to answer to the question somewhere.
Yeah, thank you, Annarita. So I remind everybody that you can please write your questions in the chat. We have already some there. I will read them. So there's a question asking if gene correction can be used with all adults and children. All have the same possibility to get this cure?
So I will start answering. So the Casgevy treatment, it's true, maybe Laure can add something. It has been tested in patients that are 12 years old or older. And I think at the moment there is not a limit in the age, but it's recommended to treat patients between 12 and 35. But again, Laure, correct me if I'm wrong. I have to say that there are also other good news in the sense that Casgevy just discussed in December new data on patients and on kids that are younger than 12 years old. And apparently the data are quite encouraging.
Thank you, Annarita. So if I should move to the next question, I will read it. So there's a patient that lives in Togo and asks if at the moment there are these candidates are being collected to do the clinical trials right now, or this has been closed.
So can you unmute Laure Joseph too? Sorry, because she cannot.
Sorry.
She was. So sorry, can you say it again the answer?
Yes.
Sorry, because I was answering to Laure.
And a second that I will. Okay. Yes. So the question was that if the collection of patients to be assessed in these clinical trials is ongoing right now or not?
Yes, there is a clinical trial that just opened in Necker Hospital, but it's a different technique because it's a lentivirus addition of a gene, and unfortunately we cannot include patients who don't have a French cover for health by the health system, so it is not possible to include people who come from other countries.
Okay.
In general, I think in Europe there are several countries, so the therapy has been approved, but now the second step is to be commercialized, which means that this should be really available to the patient and reimbursed by the health care system. So there are several countries in which this is possible. Now it's UK, it was the first country in which the UK agency approved this therapy, and now it is available for a certain number of patients. I need to say, not for all the patients, because as you see, there are a lot of inclusion and exclusion criteria. There is Italy, in which also there are a lot of patients with thalassemia that also can benefit. As Laure said, France is not one, for now, it's not yet, let's say, I mean, we still don't have this therapy available for the French patient. Hopefully this will be in a few months or next year. This is, I think there are ongoing discussion. Otherwise, I think there are more and more countries that are trying to implement this type of strategy. Of course, considering that still each country, even if we are in Europe, each country needs to evaluate the efficacy of this treatment to have it commercialized and available, and also, of course, the cost, which I think is one of the major limitations of this treatment.
Thank you for the clarifications. So there's another question. First thing, thank you to you for these presentations. And there is the first question that says, fetal gamma globin has two different oxygen affinities. What implications would it cause for adults?
Yeah, I think both me and Laure, but Laure has seen already the patient, so maybe if
we're not sure, but for people who have a very high expression of HbS, the level of hemoglobin is higher, around 16 or 18 grams per deciliter in these patients, because the affinity of the oxygen is increased with IPF, so oxygen have more difficulty to be free, so the level of hemoglobin is higher. We don't know if the level of hemoglobin of 12 or 13 obtained by gene therapy is sufficient to have a normal oxygenation of tissue, but the actual results are really reassuring. So we have to wait sometimes to be sure that there will be no impact of organ function, but the effect is really good on vaso-occlusive event and on anemia for all the patients.
And then there's another question that says, why don't we knock out the distributed beta globin? The disrupted beta globin, sorry, since it could cause clotting. I think this was actually mentioned, right? The possibility to disrupt the gene.
Laure mentioned really a clinical trial that is still in the, it's a phase 1-2 trial. So it's a trial where we're trying to demonstrate that the therapy can work. So we are not yet arrived to approval or commercialization, but with Laure and Marina Cavazzana, also with the help of Mario, we are starting a trial in which really one of the goals is to increase the hemoglobin, the good hemoglobin, but the other goal is to reduce the level of the sickle hemoglobin, because also in some Cas9 patients still, I think still the level of the sickle hemoglobin is quite, is relatively high. But I think at the same time, you need to increase the good hemoglobin and reduce the bad sickle hemoglobin. That should be really the goal for sickle cell disease.
Okay, I will go to the next one. How does Cas9 selectively increase fetal hemoglobin expression in erythrocytes without disrupting BCL11A function in other cells at the molecular and regulatory level?
This is because what I try to explain is that there are different elements that promote the expression, the production of BCL11 in different cells. There is one element that is important for the production of BCL11 in red blood cells, and another element that is important for the other cells, like the cells of the immune system. So Cas9 again is smart enough, because this therapy was developed several years ago, just to inactivate the element that is active in red blood cells. So basically, in the cells that are not red blood cells, in the immune cells, there are all the components that can still assure the expression of the BCL11 gene. So this is fundamental for having a good immune system to keep BCL11, but it's just a matter of targeting the right button in red blood cells versus immune cells to turn off a gene.
Okay, I think there are two more questions, so we have maybe 1 minute to answer. There is a patient asking if the new hemoglobin is F when the patient gets the treatment.
Yes.
And then
for beta thalassemia, it's more than 80% of HbF, and for SCD patients, it's around 45% of HbF and other HbF.
Thank you, Laure. And then you said that in Europe, the EMA approved the Cas9 therapy. Is it in the context of a compassionate use or medical need program?
I just responded in the chat. It's a medical need program in some countries, UK, Italy, Germany, Belgium, Austria, Denmark. But in other countries, it's still in discussion for the reimbursement, included in France.
Thank you, Laura, for the clarification. So that were all the questions we got in the chat. So I think now it's time to close the webinar. And again, thank you, everybody, the speakers and the listeners and the registers to actively connect and ask the questions and answer any doubts. And we look forward to see you in the next webinar sessions coming in 2 months.
Thank you very much. Thank you.
Thanks.
Thank you. Goodbye.
Bye.